Is the recording started? Yep. All right. Good morning, everyone. Thank you for joining us in today's Cambiowebinar.
Monica, we are not seeing you.
No?
No. Yeah, move to your left.
Oh, because you're here? Yeah, okay. So, good morning, everyone. Thank you for participating in Cambiowebinar. Today, we have a small workshop. The title is Accessing Genomes with Ensembl. And Emily Perry is going to talk to you about that. Thank you, and I'll leave you with her.
All right.
Hi. So I suppose it's good morning for you. My name's Emily Perry. I'm part of the Ensembl outreach team, which means that my job is to, in various different ways, teach people how to use Ensembl. So I do quite a few webinars. I do a lot more workshops in person. I work on the help desk, so if you ever email us asking for help, I might be one of the people who replies. What else do? I write some of the help and documentation that you see online, and I'm also involved when we develop sort of new tools and things. I'm involved in making sure that they're nice and intuitive and easy to use. So lots of different things, quite busy. So what we're going to talk about today is we're going to talk about Ensembl data. We're going to talk, and that will cover our genes and our transcripts. Oh, I'm getting a bit of an echo there. I'll also talk about genetic variation, and I'll talk about gene regulation, and I'll also talk about getting help. Please do ask questions during the talk, although I might encourage you to come nearer to the microphone, and there'll be plenty of time for questions and discussion at the end, and if there's anything extra you would like me to show you, I can do that as well. So we'll start with talking about what genome browsers are and why we need them. So Ensembl is a genome browser, and they're really important, they're really useful because what we seem to be producing a lot of in modern science is genomes. And we've gone from producing 10 years to produce the human genome sequence back at the beginning of the century to now producing human genome sequences in less than a week. And we're producing just vast amounts of sequence data, which in itself is absolutely meaningless. You have to process it, and you have to turn it into something that biologists can actually use, which is what we're aiming to do. And so we are a genome browser, but there are other genome browsers out there. The three major ones are ourselves. We are Ensembl.org. There's also the UCSC genome browser, which is produced at the University of California, Santa Cruz. It's a very nice piece of software, and they have lots of nice data, and they work hard on maintaining it. It works slightly differently to Ensembl, but it's still pretty good. And there's also NCBI Map Viewer, which is integrated in with a lot of the other resources that NCI produce, such as RecSeq and DBSNP and things. So we have quite a lot of data that we put into Ensembl. So we have what we call gene builds, and we have these for about 70 species. For all of the genes that we produce, in these gene builds, we compare every single one with every other one. And from this, we produce gene trees, and then we can infer our orthologs and paralogs from these. We also have what we call a regulatory build. So we incorporate data in from a variety of different sources, including ENCODE, in order to predict where there will be things like enhancers, promoters, insulators in the genome. We also have lots of different ways to display variation data, and we have a tool called the Variant Effect Predictor, or the VEP, which you can use to determine the effect of your own variants if you've done an experiment. We have lots of different ways to display your own user data. We have a tool called Biomart, which we use for data export. And we also have programmatic access. So apologies if this comes out as complete computer jargon. You probably don't need to know it, but we do have a MySQL database, which you can access programmatically via our Perl and REST APIs. Everything that we have is completely free and open. Every piece of software Every piece of code is open, is free. You can download it. You can build your own Ensembl in-house. You can change it to whatever you want it to be. It is completely free and completely open. So we cover vertebrates in Ensembl. We have about 70 species, and we try and cover a wide range of taxonomic space. We also try and cover species that have some kind of importance. So we cover model organisms for medicine. We cover agricultural species. We cover conservationally important species. So we really try and cover as much as we can with the resources that we have. If you're eagle-eyed, you may have spotted these over here, which are not vertebrates. So we have C. elegans, Drosophila melanogaster, and Saccharomyces cerevisiae. These are in there for a couple of reasons. The first one is historical. They've been in there for a long time, and when we sort of changed things, we kept them in. The second reason is for doing our gene trees. When you produce a gene tree, it's really important to have an outgroup, which you can then use to anchor the tree. So we use these kind of out species as our tree anchors. If you don't work with vertebrates, if you work with other kinds of species, you might be interested in our sister site, which is ensemblegenomes.org. Works in exactly the same way, but it focuses on different parts of taxonomy. So there are 5 mini-sites. We have bacteria, protists, fungi, metazoa, and plants, which are all available. So I'm going to jump out of the presentation now and do a little bit of hands-on. And we'll have a look at a region of the human genome. And I'd just like to show you some of the features that you can see when you look at a genome and how you can manipulate the image. So to save time, I've already navigated to this region. So this is a genomic region in Ensembl. We're on chromosome 4, so you can see where we are. And there's also, you can see there's a chromosome at the top. And our position is shown here in this red box. We have 2 more displays in this view. So we have this kind of overview of the region we're looking at. This is a megabase wide. So depending on which species you're looking at, you'll have overviews that are different sizes, depending on how dense their genes are and how big they are. And this is the region we're looking at in the middle. For some reason it's not, there we go, there's my red box. And because I moved around, it's then asking me at the bottom if I want to reset or update. So I'll just reset back to where I was. So this red box is showing us where we are. And here are the genes shown as these big gold boxes. If I scroll further down the page, you can see this is our detailed view of the genome. And this is the region we're looking at. So in the middle here, we have this blue contig. These are the chunks of sequence that the genome was made up of. We have genes which are shown both below and above the contig. Now the ones that are shown above the contig, these are on the positive strand, and the ones that are shown below the contig are on the negative strand as well. We have a legend at the bottom showing us what all the colour codes mean. We also have some features which are non-stranded. So we have things here like there are conserved elements, so conservation. These are from alignments between many species. And at the bottom we have some variants and also some regulatory features. These features are non-stranded, which is why they're shown either at the very top or the very bottom. So anything that's stranded will be shown close to the blue bar and either above or below depending on its strands. This is a view that we can manipulate. We can change it to show the different things that we're interested in. And you can do this using this blue button here, which is configure this page. Oh, it's telling me I have an incoming call. Okay, sorry about that. So if I click on this blue configure this page button, I then get a menu with all kinds of different features that I can use to manipulate this view. So I have a menu down the side with all sorts of things that I can add. Sorry, these are the tracks that I can currently see. So you can see they've all got names, and they've got these little info buttons beside them where you can get descriptions of them. But you can add other tracks using these different categories down the side. So I can click on, say, variation, and I'll have a bunch of tracks related to variation. So I can turn on, say, sequence variants from dbSNP, and I'll just put that in the style normal. But I can also turn on tracks. I've got a button here I can do find, and I can put in a word, say protein, and I'll get all the options of protein tracks as well that I can add. So I'll put on these proteins from Uniprot in normal as well. And when I close this, which I did by clicking on the tick, you'll see that I get a bunch of extra stuff. So here are my proteins, and here are my variants. So this is a view that I can manipulate to see the things that I'm interested in, to be able to look at different regions of the genome. I'm going to go back into the presentation now, and we're going to look briefly at genes. Just wait till that disappears. Right. So we saw genes a little bit in that view, and I want to describe exactly what it is that we can see. So this is how we portray genes in Ensembl. This is BRCA2, and as you can see, there are 6 transcripts of this gene. And there's a few things I'd like to point out that you can see. You can learn about these different genes, the different transcripts of this gene, just by looking at this display. The first thing you can see is the structure of them. So we can see we have these filled-in blocks, which are coding exons. We have empty blocks, such as in this UTR at the end, which is a non-coding exon. Linking the exons together, we have these lines, which look like this, which are our introns. We also have, if this gene is on the positive strand, which we can tell because it's above the blue bar, if we're on the negative strand, our introns would look like this. But we already know it's positive strand because it's above the blue bar. And another indicator that it's positive strand is this little arrow here, which indicates the direction of transcription. The colour codes have a lot of meaning as well. So we have 3 colour codes shown here. So this red one is a protein coding transcript, as is this red one down here. These blue ones are also a non-coding, and this gold one is also protein coding, but it's special. It's what we call a merged transcript. And when we talk about merged transcripts, what that means is we've used 2 different methods of annotation, and they've both come up with the same exact transcript. So the 2 different methods we use are Havana manual annotation and Ensembl automatic annotation. Now, anything that's been annotated by the manual annotation has a number that starts with a 0. So all of the transcripts are named after the gene, followed by a dash and then a 3-digit number. If it starts with a 0, it was annotated manually. If it starts with a 2, then it was annotated automatically. So we can see that this gold one, this red one, and all of these blue ones have been manually annotated, and this red one has only been automatically annotated. So these golden transcripts are identical annotation between 2 different methods, and we have this for 5 different species. We consider these to be high confidence and quality. So we have 2 different methods of annotation from Ensembl and Havana for different numbers of species. Automatic annotation for Ensembl is done for every species in the database, whereas Havana manual annotation is only done for a smaller number. So the automatic annotation is done using a computer pipeline. We basically take the complete sequence of the genome, and then we take a whole bunch of gene models, which we get from different databases, and we plot them onto the genome. And where they fall on the genome, based on sequence, is where we put the genes. One of the misconceptions people have is that they think that we predict genes from open reading frames. We do actually do that, but they don't make it into the gene set. So we do it, we use it as a check to make sure everything's working, but that's it. We don't touch them after that, and nothing will go into the gene set unless it has real biological data behind it. So the data that we use, this real biological data I'm talking about, comes from a variety of different sources. So we use the INSDC, which is the International Nucleotide Sequence Database Consortium. That's made up of ENA, which is basically EBI with me, from GenBank, which is at NCBI, and from DDBJ, which is in Japan. And these are databases. If you get some kind of biological sequence, you can submit it to one of these databases. They will then share between each other, so every 24 hours they do a data share. And then, so that means that if you ever look at any one of them, it should have almost identical data to the other 2. And so we take nucleotide sequences from those. We also use protein sequences, so we use UniProt. And UniProt has two databases within it. They have SwissProt, which is manually curated, and Ensembl, which is unreviewed translation. We use both of these. And then we use NCBI-RefSeq, so we use the manually annotated proteins and mRNAs. These are the ones that begin with either NP or NM. And so we take all of these and we plot them onto the genome, and from this we determine where the genes are. Manual gene annotation is a bit different. This is done, by a person rather than by a computer. And so somebody will spend maybe half a day on each gene, plotting the sequence onto the genome, and then really paying attention to things like splice sites, like untranslated regions. And this is done by the Havana Group, which are based in the Wellcome Trust Sanger Institute, who are on the same campus as us. And they're able to produce genome-wide data for human, mouse, zebrafish, pig, and rat. And they also produce less extensive data for a few other species. They have their own browser for this, which is called Vega, where if you're interested in just manual gene annotation, you can take a look. They're able to use not just databases, but also things like papers, and they can evaluate the quality of the data as well as sort of putting thresholds on it, which is all you're really able to do with annotation, manual, automatic. So you might have heard of a set of genes called GenCode. So GenCode is actually made up of the Ensembl automatic annotated genes, the Havana manually annotated genes, and what we call the merged set, which is wherever we get a perfect match between the two methods of annotation. GenCode came about because the ENCODE project wanted to have a gene set, and they realized that creating their own we'd spent 15 years on it, so they didn't think that it was something that was easily accomplished. So they decided that our gene set was very good quality, and they took what we had, they added a few extra bits to it, and basically called it GenCode. It's ever so slightly confusing because it's the same gene set as you see in Ensembl, but we only call it GenCode when we're talking about human and mouse. The zebrafish, rat, and pig genomes are made and annotated in the same way, but for some reason they're not called GenCode. I think it's just to confuse everybody. So GenCode is used by ENCODE, it's also used by 1000 Genomes, and other projects are taking it up as well. So I'm going to have a quick look at a gene, and we can see the kind of data that you can get when you look at a gene in Ensembl. So the gene we're looking at, I've already found it here, is called ESPN, and we can see the name of the gene. It also has an ID, which Ensembl assigns to it. This ID is stable and should last between different releases, even if the gene name changes. There are loads of different features down the side that we can explore about the gene, loads of different things that we can see. We can also see a graphical display of all its different transcripts. So we've got one big, long merged transcript, and we've got a few more protein coding and some non-coding as well. There's also a table summarizing this. If I click on Show Transcript Table, you can see that I've got a list of all the different transcripts. And they've got some tags added to them as well. If you hover over them, you can learn a bit more about the kind of data quality for the different transcripts. But you can see there's lots of things I can explore. For example, I can have a look at sequence. Is that loading? Maybe I didn't click. Oh, it's doing it now. So as you can see, we can look at sequence, but there are also lots of other things. I won't go into any of them in any detail, but we can see it in other databases. We can see gene trees. We can see tables of the different variants in the gene. So there's lots and lots of data available for a gene. I can also visit an individual transcript. So if I go into this first one at the top here, you can see that I also get some sequence views for the transcript. So the cDNA view is quite nice. I'll just open that up. Here we go. So you can see we've got the sequence of the cDNA as well as the amino acid sequence of the protein product as well. And there are things like I can find out the function of it via gene ontology. Again, I can see variation, and I can also see things like the domains and features in the protein. So there's lots of things that I can explore about a transcript I'm interested in. We'll go back in to the presentation because I've got lots of things to cover. And I'd like to talk a bit about variation data. So we have variation data for a number of species, not for everything because it all depends on what is actually out there and what kind of data people are producing. And also in some of the Ensembl genome species. We get data for our variation from a number of different sources. Most of it comes through dbSNP, but we also get extra things like we get phenotypes from the GWAS catalogue, we get cancer variants from COSMIC, we get structural variants from DGBA, as well as many other sources. There is a URL here. Somebody should have a copy of the PDF, and I'm sure that can be distributed to you so you can get these URLs. So we have small-scale variants, things that affect only one or a few nucleotides, or large-scale variants, which structural variants such as copy number variation, large-scale deletions and duplications and insertions as well. In terms of the small variants, we categorize them based on how they affect the genes they hit, so where in the genes they fall, and what kind of, whether or not they change the coding sequence in the genes where they fall. If they do change the coding sequence, we use these two algorithms called SIFT and polyphen, which are scores for the change in the amino acid sequence. These are based on two things. Firstly, how well conserved the protein is. So it's considered that a highly conserved protein, if you change amino acids in that, that's probably going to be a bad thing. But if the protein is very different in every animal you find it in, then it's probably not so much of a big deal if you change an amino acid. It's also based on chemical changes in the amino acid, so changing from a neutral to a neutral amino acid is not a big deal, but changing from a positive to a negative is quite a big deal. So SIFT and polyphen both score out of 1, and really helpfully they clearly did not communicate with each other at all because their scales are completely opposite, and I never remember which one is which. This is why I make slides that show me. So SIFT say that anything that with a score of 0 is really bad and your protein is going to be completely destroyed and isn't going to work, and 1 is your protein is perfectly fine and don't worry about it. Whereas polyphen say 1 is your protein is destroyed and 0 is your protein is perfectly fine. Fortunately, they also produce predictions, so they have cutoff points, and they will label anything at certain cutoff points as being deleterious or tolerated for SIFT, or probably damaging, possibly damaging, or benign for polyphen. These are the colours. The colours on this slide are the colours that we will use to label them in Ensembl, but if you ever hover over them, these scores, you'll also get the prediction. So if you're colourblind and can't see that, then you'll also get the words as well. You might be coming in with your own variation data rather than just looking to see what data we have about variants that are already known. And if you've got data in a variety of different formats, you can actually use it in our tool, which is called the Variant Effect Predictor. And what the Variant Effect Predictor does is it gives us the consequences of all the variants that you've put in, whether those variants already are known or not, and so it's able to see which genes they hit. We have this available as a web interface. It's all point and click, and you can copy and paste stuff in. We also have a standalone Perl script, which we have extensive documentation on how you can use it and what commands you would need to type into the command line to make it work. So it's a really nice piece of software. It is our most popular tool. We've got lots of people copying it now. So when we first brought it out, it was sort of new and innovative, and now there are loads and loads of people making similar tools, which I think is the best mark that it's a good piece of software and it fills a need. So I'm just going to show you briefly what you can see if we look at a particular variant. So we're going to look at this variant. This is a dbSNP ID. They begin with RS, and we'll just briefly look at some of the data we can find out about a variant. So this is our ID, and we've got these lovely pretty buttons that link to different pages that we can find out about it. We can see that the alleles are G/A, the ancestral allele is G, and the minor allele is A, and its frequency globally is 0.23. If I were interested in more fine-grained population genetics, I could click here, and I could find out its frequency in different populations. And these are data from 1,000 genomes. You can see that in the Asian population, the G allele is 100%, whereas in the European population, the A allele is actually slightly more common. I can expand these out, see more data, and I can also see breakdowns of the numbers in the different populations. I can also explore phenotype, which is quite useful, and so I can see that this particular variant is involved in lactase persistence, so the ability to digest lactose as an adult. And I can also see which genes it hits. So if I go into genes and regulation, I can see that it falls in a gene called NCM6, and it's intronic to all the different transcripts. It's actually non-coding. 2 of those transcripts are actually non-coding. And there are more things that I can explore, but we don't have huge amounts of time, so I'm going to move on. Okay. So I'd like to talk a bit about regulatory data. So in Ensembl, we're trying to work in kind of epigenomic data. So epigenetics is known to regulate gene expression through heritable changes in the gene and in the genome without actual changes in DNA sequence. When we talk about epigenomics, we basically mean the same thing, but we're talking genome-wide. And this is a very new branch of study. It's in its very early stages. And so what we're trying to do in Ensembl is we're trying to annotate the genome with features that can play a role in the transcriptional regulation of genes. And we're looking at a lot of different kinds of features. So we're looking at predicted open and closed chromatin, which we find out through DNase 1 sensitivity and FAIR studies. We look at transcription factor binding sites from ChIP-seq. And we look at the epigenetic marks, so we get histone modifications, again from ChIP-seq, and DNA methylation from bisulfite sequencing. And we also use RNA polymerase binding, again from ChIP-seq. And we process, these data come from a number of different sources. So at the moment in human, we have data from ENCODE and from Roadmap Epigenomics. And we've got blueprint data as well in the pipeline, waiting for us to work with it. And we don't just use the raw data, we process the data. So we don't just say, here's where all the histones are binding. We use that to predict where things like, we say, well, we know that when all these histones are bound together, that's normally an enhancer, or that's normally a promoter. And that allows us to be able to say, based on all of these data, here's a promoter, here's an enhancer, and you can see that in the browser. So I'm going to look at a region of a particular gene and see what kind of features have been predicted. So we're at another region here, and we can see these are the regulatory features that I showed you earlier. There is a legend telling us that these are CTCF binding sites, and this is a promoter. If I click on them, I get a little pop-up, and there's a link to a stable ID for the feature. If I click on that, I go to the regulatory feature, and I can see that of all the cell lines that have been studied in this, our promoter is inactive in some of them and active in others. So where it's shown in this hashed box, it means it's not active. And I can see that it's active in 6 out of 18 of the cell lines that were actually looked at. If I go into details by cell type here, I can see that in this particular cell line, you can actually see what were the peaks and the features that were used to construct it. So that we can see that overlapping this predicted promoter, we have a region of DNase 1 sensitivity, we have polymerase 2 binding, and we have H3K4 trimethylation. So we can see that there are different features in the different cell lines. I can also choose to show extra cells, and it's only showing me some of the available evidence here. But there are extra things available that I can add if I want to learn more. Go back into the presentation. For now, I think I'm a little bit ahead of time, but that gives us more chance to have a look at some of your questions. So I did want to go over a little bit on help and documentation. So we have online courses available. There's a brilliant site which is called EBI Train Online, where there are loads and loads of online free bioinformatics courses that you can have a go at. We have loads and loads of tutorials on our website. We have a YouTube channel with loads and loads of short videos which take you through different kinds of features. It'll be similar sorts of things that I showed you in the demos, but they'll be much more detailed, they'll be much longer on a particular topic. If you're using Ensembl and you get stuck with something and you want to get help, you can email us. We are helpdesk@ensembl.org. It'll be me or one of my colleagues who will get back to you and say, oh yes, that's happening because of this, or oh, that data needs to be fixed. Thank you for letting us know, whatever it might be. We also have a couple of public mailing lists. So if you're using programmatic things, you can join our dev mailing list and you can communicate with our developers, but also with other people who are working with the resource. You might be doing something that you're trying to do. And we have an announced mailing list to tell you about new things that are coming out. We do have social media, please do follow us. And we do produce papers as well. So if you work with our data, please do cite us as well. And this is the team. These are all the people who work for us. And these are the people who pay for us as well. So we like them. I'm happy to take any questions. I realise I was trying to make sure we got everything in, and I've gone a bit ahead of time. So now would be a good time if there's a particular piece of data you're interested in, or if there's a particular kind of view that you would like to show me to show you how to use. Please do ask questions.
I have a question here.
Yeah?
Yes. I am very interested in the VB. Can you tell me a little bit more about it? What are the factors you need to take into account to predict the variant effect?
Okay, so the variant effect predictor is basically what it's looking at is where on the genes, what genes you're hitting and how. So if we look at this is basically what it's predicting. It's using the position of the variants in the genome, seeing which it matches up with, and seeing where in the genes it hits. So if it falls within a coding region, we'll then ask, well, does it change the amino acid sequence, or does it not? If it falls within a regulatory region, we'll tell you that. If it falls within a motif, so like a transcription factor binding motif, we will tell you that it's fallen in that motif, and we'll give you if it's changed, the score of the motif will tell you by how much, because there are assigned scores based on how well that particular sort of 10-base sequence or whatever it might be can bind the transcription factor. So there's a lot of different things that it can predict. I can actually show you what kind of things it will give you. So the variant effect predictor is here. Just click on there. If I launch the tool, I'm going to put in the variant that I used earlier in the demo because it's quite a good one. Actually, no, it's not, because it doesn't hit gene. So it gives me some example, there's a sort of example bit of text in there. So I'll just leave that as it is. And if I run the tool, it takes a moment because it has to process these data. Now, you can do this with an entire, like a whole genome SNP calling. So you can, oh, it's a bit slower, wants to refresh again. It's refreshing every 10 seconds. So you can use a massive VCF file, for example, from a whole genome SNP calling. Right, if I view results here, so it tells us kind of which genes it hits and what the consequences on those genes are, which is quite useful. Unfortunately, this example doesn't give me any missense variants, but it gives me, though, if I had missense variants, I would have those system polyphen scores that I talked about earlier, which tell you kind of how bad the change in the amino acid sequence is. It can also, so it can come up, it will work with variants that are not already known. It will work with variants that are already known as well. And if they are already known, it will actually tell you in this column if your variants are known. And then it can give you information about them as well. So it's a really nice flexible tool that gives you lots of information.
So is there a limit to the amount of variants that you can ask at the same time?
No. So we have, if you're using the online tool, it would be quite slow. But there is a script that you can download. If you're working with more than, I would say, a couple of thousand, you're best off working with our standalone script, because then it does it in-house. You can also download a cache with all our data, so that it does it all within your own system, rather than having to communicate with our database, which can make it much quicker. But essentially, there is no limit.
Okay.
Quite slow.
And also, I know there is a lot of, everybody knows there's a lot of things that we don't know, especially in terms of the promoters. We know some motifs that are usually important, but we know there are so many other parts of the promoters that can vary their sequence, and we know very little about them. Those VP tells you something about, well, maybe, but we don't know, there's nothing sure, but it has a chance that it is causing a problem.
We don't know a huge amount either. So we're predicting where promoters are based on where we're seeing transcription factor binding, where we're seeing certain modifications. We certainly, I mean, with a promoter it's fairly easy to guess which genes they work with. But certainly we're predicting things like enhancers. We have no idea what genes they might be working with. All we can say is that they have features that look like promoters that look like enhancers that we've already seen. We're essentially putting these data out there. So when we put, when we put something like this available online, what we're saying is, we want you as researchers to then go and find out more. This is really kind of early stage data. Epigenomics has only been around for the past couple of years, really, and there's this massive explosion of data, and we really need the sort of help of the researchers in order to interpret it. So we're putting out there as much as we can in the hope that then somebody will come along and say, okay, let's say this histone modification that we can see in this particular location, let's investigate why it is having this particular effect. I can't see that many. I'll put on some more transcription factors so we can see a bit more. Now I'm going to, so we can see that we've got this transcription factor here, seems to coincide a reasonable amount with this histone modification. We don't know why, and we're really putting it out there so that biologists can do the experiments and investigate this and work out what's going on.
Okay. If I have questions on what I'm looking at, for example, I can look at that picture and you explain what the colors mean. But is there a feature that I can ask, hey, what are we looking at right now?
Yeah. So most of the pages have this little I button. So if you click on the little I button, it generally gives you a description of what's going on in the page. Most of them will have a legend at the sides of these views, so you can make use of the legends. And if you ever think that you can't find it, that it's not obvious on the page that you're looking at, that is when you send us an email, and you can ask us what's going on if you're ever confused, and we will try and get back to you in about 24 hours.
Okay.
Unless it's a weekend. Or Christmas or something.
So I I have another question. You probably have some exercises so that students can perform those exercises and get familiarized with the different resources.
Do you want them? Do you want them? Shall I send them to Monica and have her pass them on to you?
Yeah, I think some students will benefit from them.
So I can send you my kind of standard course book, which is a bit more extensive, a lot more extensive than what I've covered today. So there's a full course online which has got loads of exercises in it. But I can send you my up-to-date course book, which has got little walkthroughs to take you through different bits of data. We'll look at similar things. It looks at similar things that we've looked at today, but in a lot more detail, and explains kind of what they are. And then we've got exercises where you can practice using them. I will send that to Monica, and she can share that with you, and then you can all have a go at that. The course that I'm talking about would take about a day to do in person, so it's not a small course. But you can dip in and have a go at different things that you're interested in.
Okay, well, thank you very much.
Well, thank you for inviting me. This is lovely. It'd be much nicer if I could come and visit, but never mind.
You never know.
Thank you everyone for participating.
Okay.
I'm going to send you a link with a feedback survey. If you can take a couple of minutes to fill it up, it will really help us to tailor these seminars to your liking. So, thank you very much. And right now, the recording is going to take a little 50 more minutes. So, since we are done early, it's okay. We can try to disconnect somehow. And then that's it for now. Thanks to Emily so much.
Sure, thank you very much. I hope you repeat next semester.
All right.
Thank you.
Bye.